Good morning, everyone. I'm very happy to be here today, and I will try to bring a biological view today to the question that all of you are asking about how life experiences influence our body, our health, but I will also bring not only a biological understanding of this, but how our life experiences can also influence our children, grandchildren, and perhaps even further. So Carmen asked me to say where I come from scientifically or professionally speaking. So I was trained as a molecular biologist and behavioralist, so to ask fundamental questions about brain functions, how the brain works. And initially I worked on memory because I was interested. As she said, I did a postdoc, I did a PhD in Basel and then a postdoc at Columbia University, and there the major question was, how do we forget? How does the brain forget information that we experience? And what are the mechanisms in cells which explain forgetting and the failure in memory formation in aging, for instance? While I was working on this, that's a super interesting research, very competitive research, but I thought I was getting more and more interested into the bigger picture. How life experiences can really modify our memory, but can modify our whole body, who we are, basically. And at the time, the notion of epigenetic was not really considered in neuroscience or even in medicine, because it's a totally different way to think about the body, to think about how cells and biology works in the body. You have, I'm sure, all the idea that we are what our parents are. So there is genetic. You inherit half of the genes of your mother, half of the gene of your father, and that's your genes, your genome, which makes who you are. And this is still an idea which is somehow dominating, but now what the research in epigenetics has provided new or has complemented this view is that it's not just the genes which are important for us, but all our experience, all the things we are exposed to during our life can also modify the genes. Not the sequence, because the genes, it's a bit, if I take the homology of a computer, our genes is a bit like the hardware of the computer. You know, it's something solid. It's a code that we cannot really change. We can damage the code if we are exposed to X-rays, to UVs, to carcinogens and radiations, but we cannot, besides this, we don't modify it. But the epigenetic is all the, all the mechanisms which read the code, because a computer has softwares. You need the softwares to read the hardware, because the computer alone without softwares is useless. Your genome alone, without many, many millions of mechanisms or proteins around it to read it, interpret it, this genome is not worth anything. And it's the environment which modifies the way the genome is used and which makes what we are indeed. I titled my talk, The Power of the Environment on Our Genome. So I will try today to really explain the concept of the importance of considering the environment for understanding how the body works. I think through your profession, I'm sure you are all completely convinced that the life experiences, what you have, how you grew up, the environment during childhood, whether it was positive, rewarding, or traumatic, negative, neglect, with violence, whether physical or sexual violence, that all these experiences can have a very, very strong impact on your body, on your mood, on your mental health, on your physical health, and that everything is intertwined, and that it's very difficult to distinguish what effect, what symptom comes from where, and how things interact. So from a biological perspective, it's also extremely complicated to explain this, but we are starting to gain understanding. So the major concept is that there are many factors in our brain which influence our brain and our body throughout life. Here we have an adult, but it could be a baby, a young boy, a young girl. Throughout life, really, what we eat, whether positive or negative, diet, what we experience, whether positive experiences or stress, trauma, in particular in childhood, drugs and chemicals we ingest or we are exposed to throughout our life, environmental toxins, pollution, endocrine disruptors which are in the environment, which we are exposed to every day through many different ways, plastic, food, which is not organic, but also the lifestyle, whether we practice a sport, whether we have a rich and dense social network, whether we have a rich family, a dense family and a happy family. All these factors together, alone of course, but also together can influence our body. And basically every single cell in our body can be modified. Brain cells, heart cells, but also germ cells, because germ cells, which are sperm cells or germ cells in development, which are going to give sperm cells or oocytes, which will, after fertilization with sperm cells, will give an embryo, can also be affected by any factor we are exposed to during our life. And that's really the key of the concept. If we want now to question not just the effect of the environment or our life experiences on us, but on our children and perhaps our grandchildren. Of course, there is always the cultural transmission. If you are nice and sweet and caring with your child, there is a higher chance that your child is going to be a nice person, a balanced person, and that your child is going to have a happy life. If, on the contrary, you are a violent person, you don't take care of your child, or in general, a child is not well taken care of, there is violence, emotional pressure, this child has fewer chance or higher risk to not develop properly, to not have a proper self-esteem, and to have herself or himself psychiatric problems or even physical problems later in life. This is cultural. But now I'm talking about the biological inheritance, what happens in the germ cells, in sperm cells, in oocytes, which in addition to genes can be transferred to the future generations. And that's the novelty of the concept, is that it's really in addition to the code, which you cannot do anything about, it's just from both parents, it's there and it is transmitted, exactly like it was given by the parents. But the epigenetic factors, those factors which are around the DNA, which are influenced by life experiences, it's the factors which can also be transmitted and can explain some of the perpetuation of the effect of life experiences. Now, if we think of this notion of germ cells being affected and how much transmission this can have for future generations. Now, if you take a man, it could be a young boy, so if let's say this person is exposed to endocrine disruptors or to a very severe trauma in childhood, not just all his cells can be affected, but also his germ cells, so his future sperm cells. And if there is something which is modified there and which remains modified, the risk that it is passed to the next generation, to the children, is high. Now, if we take a pregnant mother, so it's not just all her cells, but all the cells of her baby, but in the baby there are also future gametes, cells which will become sperm cells or oocytes one day. And if this woman is exposed to endocrine disruptors through diet or through, if she lives in the countryside and the farmer next to her is spreading pesticides on fields, and if she eats a lot of food which stays in plastic for a long time, all these multiple factors, maybe her will be affected, her baby, but also her grandchildren, if these gametes are also affected. And the risk is high because these gametes, they are cells, undifferentiated cells, which are exposed through the blood of the mother. And they are exposed for a long time, for weeks and months. So this means that there is a risk that it may be passed to the third generation. Now, it could be, this concerns any type of exposure. It could be that the further generations, fourth, fifth, in if, let's say, we assume that once the gametes here, or the sperm cells, they are affected, if the alterations stay for a reason we don't yet understand exactly how this could be, but we start to have evidence that this may be the case. If there are changes which stay, maybe further generations could even be affected. What are the evidence in nature for this type of inheritance? So this is called epigenetic inheritance because it's beyond genetic. So genetic inheritance is the inheritance from the genes, from this code, which is very well known, very specific for your mom and your dad. Genetic inheritance. It's clear if you sequence your genome, you know exactly what the genes come from your mother and those which come from your father. This is now epigenetic inheritance. So there are all the additional factors beyond the genetic code. So there is lots of evidence in invertebrates that epigenetic inheritance happens. In C. elegans, so C. elegans, it's a little worm like this. It's a model which is quite heavily used in research because it's nice, it develops, it breeds very easily, it's very small, so you don't need big labs to keep them, and they are quite easy to study. And there are many labs in the past 10, 20 years which have demonstrated that if you starve a worm or if you expose the worm to a hormone or whatever, that it can induce symptoms which are transmitted across multiple generations, here and down to the 14th generations. In mice, which are other models, which are closer to humans because they are mammals, and in the lab they are extensively used. It's a nice model because it breeds quite well. The gestation is quite rapid, it's 3 weeks. So you can generate multiple generations in mice. It still takes a couple of years to generate offspring, but another advantage of rodents is that, or lab mice, is that their genome is completely known. It has been sequenced, and it's to work as an instrument. There is a lot of information that we can use to understand the symptoms, for instance, of exposure. And in mice, recent research, including ours, showed that postnatal trauma or paternal obesity or drugs like valproic acid or endocrine disruptors can have symptoms, physical symptoms or symptoms on memory or behavior, which can be transmitted across multiple generations. And we have shown that postnatal trauma in mice can have effect through the 5th or the 6th generation through the father. If it's the fathers which are used to breed, to generate the next generation, and symptoms of risk-taking, for instance, are quite prominent till the 5th generation. Now, in human, there are also many epidemiological studies, so studies on very large cohorts in the context of stress, like Rwanda genocide or Holocaust, or parental separation, or famine during Second World War, for instance. And many of these large studies, some of them include several thousands of people, they show that indeed exposure of parents or grandparents can explain, correlate with symptoms, metabolic disease, for instance, like obesity, diabetes, or even increased mortality in the children or grandchildren. But in humans, unlike in rodents, it's only correlative. You know, in rodents, the way we work is that we have male and female, we breed them, we generate pups, and we control everything of their life. So we know that if these mice, they are exposed to traumatic experience and no other experience, we control what they eat, what they do, and the cages and everything. So if they show symptoms when they are adult, or if the children or grandchildren show symptoms, we know that it is due to the experience that the parent or grandparent, father or grandfather has seen or lived. In human, it's a lot more difficult to really associate like famine in great-grandparents and diabetes or cardiovascular disease in the descendants, because human life is complex and there are multiple factors which can intervene. So we can talk about inheritance of the effect of life experiences in human, but we cannot be 100% sure that it's of the mechanism. What are the mechanisms responsible for this transmission? Research at the moment is still really at the beginning to look, for instance, at germ cells in humans who have been exposed and see whether there are any changes in germ cells which could explain transmission of these effects. Now, just a few examples of evidence that some exposure or life experiences can have effects across generations. The best known experience or exposure is diet. So it's known that diet influences the health in the offspring. So it's been shown in different cohorts. One of them is an Överkalix study. Överkalix is a small village in northeast of Sweden where people were, the study started with people born at the end of the 19th century, and there were studies done in their children and grandchildren. And it's not related to any war, but it's just a very rural village where, by looking at the archives of the village, people could evaluate the caloric intake of these people. And what these studies, which were performed by people in Sweden, researchers in Sweden, showed that when there were a lack of caloric intake in the grandfathers or grandmothers, when they were themselves adolescent, this had a negative effect on the children and grandchildren. With them, their life expectancy was shorter, and they had increased incidence of diabetes and cardiovascular diseases. This was confirmed by a more recent study in Uppsala, in Sweden as well, where this study is even larger. It's more than 10,000 people. So confirming that what your grandfather or grandmother ate, the diet in particular during adolescence, can have very concrete and very strong consequences for your own health. In terms of nutritional insults, there have been also many studies on cohorts linked to the Second World War, because at the end of the Second World War in West Holland, there was a big Nazi embargo, and people suffered for a couple of months very strong famine. And it was found that in the children and grandchildren, there is lower weight at birth, and also an increased risk for cardiovascular disease, hypertension, and other diseases like diabetes and obesity in the second and third generation. And this has been confirmed across multiple studies, so which is again, evidence that diet is one of the major factors that can influence health in the descendants. Now, in terms of other experiences, which don't involve any intake of food or toxics, or which is more emotional and psychological, it's very well known that traumatic experiences can affect people quite severely, in particular childhood trauma. So violence, abuse, violence, whether physical or sexual, neglect, all these detrimental conditions in childhood can often lead to depression, antisocial behaviors, borderline personality disorder being one of the diseases, which is complex disease, which includes risk-taking, cognitive problems, antisocial behaviors, and a tendency to commit suicide. So this is in borderline personality disorder patients. Most often, people have experienced violence in their childhood. So it's known that traumatic experiences in early life, in early childhood, can increase the risk to develop such diseases, but these diseases are very comorbid. So often someone who is depressed may also have cardiometabolic alterations or have a higher susceptibility to develop cancer. And what's very prominent also in this type of diseases is that it's known that they run in families. And there have been many, many genetic studies, genetic association studies, to try to identify the gene for depression, the gene for schizophrenia, the gene for these complex diseases. And up to now, it's known that there are susceptibility factors, like small mutations on the genome, in the genome, which may increase the risk to develop these diseases, but it's also acknowledged that it's not just genetic. It's a combination of genetic susceptibility and epigenetic factors. And indeed, I believe that the epigenetic factors, which is the environmental and life experiences, can be extremely, can have an extremely strong role in the development of these diseases. In mice, for instance, if we take mice which are all genetically identical, it's called isogenic. They all have the same genome. And we take a few of them and we expose them to traumatic experience. It is sufficient to induce a whole lot of symptoms across multiple generations. So which suggests that the genome, the genes, are not really involved there. You don't need genes to be different in mice. It's not like some have a more susceptibility because they have a different genome. If they all have the same genome, you expose them to trauma, they will all develop symptoms. So I think in human, it's not known what percent or how much the genetics influence the development of psychiatric disorders over life experiences, but I think life experiences can have a very strong role and very strong impact in humans. As I said, there are many, many factors, and now it's a lot more documented, not only in animals, but also in humans, and there is a review which compiled all the dozens of studies published recently. Now, in terms of mechanism, we want to understand what is it, how is it working? How can the genes or the genome be influenced by diet, by life experiences, by traumatic events? Our genetic makeup, so we are constituted of many, many cells, and cells have a nucleus, and nuclei have chromosomes, so we have 20 pairs of chromosomes. And chromosomes are made of the DNA. So if you pull on the chromosome here, I will show you next a small video for you to picture everything better. So you have here the DNA, so it's a long stretch with a specific sequence, and here it's illustrated by, so the sequence has 4 bases, and these bases are placed in a specific sequence, and each of us has a specific sequence. So that's the genetic code, if you wish. So you have the string here with these proteins, and it's all twisted onto itself to form the chromatin, which is compacted to fit into the chromosomes. And the DNA here, the code, is very important because it will be read and it will be used to produce proteins, which are the building blocks of our body, but also proteins which are necessary for the cells to function. If you, let's say, now you are thinking, you are listening to me, your brain is working, is processing the information, you need your neurons to function, to send signals in between each other, and this requires many enzymes. It requires the neuron itself, but the neuron has many cells, many components inside, which are made of proteins, and these proteins are synthesized from the DNA. But what's important to know is that this is the genome, but in addition to the genome, there are many factors which are around the genome. There are many proteins, many other forms of genetic molecules, and also proteins, which changes on the proteins, which make the epigenome. So the epigenome is, as I gave the example, the analogy with a computer, that would be the softwares of the computer, which read this code and interpret it. And life experiences modify the way we read the code and interpret the code. Here it's an example in neurons. So you have again the chromosome, the chromatin, the DNA, and you see here all these modifications, which are the epigenetic factors. The DNA itself can be modified, but also the proteins which are associated to it. So there are many small modifications. There are like chemical groups, chemical changes which are added here, and they can be added, removed, depending on your life experience, depending on what you eat, depending on whether you are exposed to endocrine disruptors or whatever. And there are also many molecules here in the cells itself, which are around the DNA, which modify it. And what you see here in neurons, pretty much the same exists also in germ cells, so in sperm cells or in oocytes. And sperm cells, they are the mature cells, but if you take a young boy, they aren't mature germ cells yet. They are germline stem cells. So there are cells which multiply all the time, and all the germline stem cells, all the cells throughout development until the mature reproductive cell, they also have an epigenetic code which can be modified by environmental factors. So now I'll show you this short video to, again, remind you, for some of you, or maybe for the others, explain what the DNA is good for. So this is a cell, and we are going to get into the cell, and we'll see the nucleus. So the cell has many pores. You get into the cell, so there are all the cell components which help the cell function, produce energy. This is the nucleus, and the nucleus has many holes. We enter into one of these pores, and here this is the chromosome. So the chromosome is made of chromatin. And if you pull on it, you see the DNA here, the double helix with the code, and the proteins it's associated to. So the double helix here has a very, very specific code. And here we have about 20,000 genes. But you need to understand that genes, they are only a very, very small portion of this long protein, of this long DNA. So the DNA is very long, and it's only 1%, which is constituted of genes. And genes are sequences which mean something. They code for a protein. And I'm going to show you now what it means, that they mean something, that they can be read and used to fabricate, to synthesize a protein which is necessary for building us and have cells function. So the code is in the chromosome, and the chromosomes are in the nucleus, remember, and they stay in the nucleus. It's very important that they are protected. So if you need to read this code and use this code, but this code is stuck in the cells, you need a system to read it and get it out of the nucleus to be able to produce proteins outside of the nucleus. And this is done by copying the DNA. The DNA is copied into RNA. You have heard a lot about RNA, messenger RNA, in the context of COVID, and that's exactly what it is. Messenger RNA is a copy of the DNA. And in the case of the COVID vaccine, what the vaccine contains is a messenger RNA which corresponds to the genetic code of the virus. How a messenger RNA is produced is you first need to separate the two strands of the DNA, and this is done with a polymerase, which is a big protein here. And you see that there is a system which opens the DNA and copies, read it. These big proteins read it. It's a bit quick. It reads, let me go back a bit. So it reads the DNA. It brings the building blocks here, copies, and add exactly the one which fits to the base that forms the DNA. And here you see it's an RNA, or a messenger RNA, which is synthesized in the nucleus. And this messenger, its RNA is called messenger because it carries a message which will allow to produce a protein. So this messenger RNA is very big. It's a little bit, I mean, there are sequences, parts of it which are not necessary. It's processed, and then it's sent out of the nucleus. So you see how smart the system is. The DNA, which is very fragile, stays in the nucleus. And now you have the RNA that you can work on. You can read the RNA to synthesize proteins. So this is done by ribosomes, which are proteins. Then you will use this carrying, it's an RNA, it's also an RNA which carries amino acids. Proteins are made of amino acids. And this code here will determine which amino acids is carried here. There are 20 different amino acids. And then you see that little by little, from the messenger RNA, there will be a protein which will be synthesized. And then the protein will be needed to fabricate cells, to have cells function, and so on. So that's what the genetic code, the genome, does. But in addition to this, there is also, as I said, the epigenome. There are many RNAs which are synthesized from the DNA, but which are not messenger RNA. They are just RNA. It's just a copy of some parts of the DNA. And these are very important to regulate the genome itself. They are part of the epigenetic factors. And these RNA, non-coding RNA, you can modify the population, you can modify the synthesis, the sequence, depending on what you eat, on your experience, on your social life, and so on. So all life experiences, whatever we do, how we function, can modify the population of non-coding RNA that we have in the body. So now if we come back to the concept of germline-dependent inheritance of environmentally induced traits, so the fact that experiences like those can modify the genome and the epigenome. And if the modifications happen in germ cells, in sperm cells, that this could explain how this is transmitted. So it's exactly what we have done in my lab in the past 20 years. So we were interested in the effect of postnatal trauma. So we developed postnatal trauma, so traumatic experiences in early life. We developed a mouse model of traumatic experiences, which is based on unpredictable separation of mouse pups from their mother. So they are separated anytime during the day for 2 weeks, from 1 day after birth till 2 weeks after birth. So it corresponds to about 2/3 of adolescence or childhood in human. And then we let them grow adult. So it's a quite short traumatic experience, it's just 2 weeks. We let them grow adult, and then when they are adult, we test the behavior, we test the physiology, we test the metabolism. We did many different analyses, here behavior and multi-omics, and we breed them across to generate multiple descendants. So I don't have time today to tell you what we found, but we found that there are many symptoms which are induced by this traumatic exposure, and it's a combination of memory problems, memory decline, risk-taking, depressive symptoms, antisocial behaviors. So lots of behavioral and cognitive alterations which are present in the exposed animals, but in the offspring, grand offspring. And as I said earlier, until the 5th generation, we see some of the symptoms. It's not just behavioral symptoms, it's also metabolic symptoms. So the glucose, insulin metabolism is altered, the lipid metabolism is altered, and recently we also did, through a collaboration with a cardiologist, a colleague cardiologist at the University of Zurich, looked at cardiac functions. Using echo MRI, exactly the same way it's done in human. And we observe that across generations, there is a cardiac hypertrophy, and cardiac functions are altered, which is similar to cardiac failure in humans. So we also know that bones are altered, or bones physiology is altered, with a tendency for sort of osteoporosis in the animals which were exposed to stress. So a multitude of symptoms in these animals, and many of these symptoms are present in the offspring and the grand offspring. We are starting now to understand what epigenetic factors are linked to this. We know that, for instance, that there are epigenetic factors in sperm cells, in the exposed males, which can, if we take the RNA, now that you understand what RNA is, sperm cells are full of RNA. If you take the RNA from sperm cells from a mouse, which has been traumatized, and you take this RNA, you inject it into an embryo, and then the embryo, you transplant it into a female, and you obtain an offspring. We found that this offspring, which is derived from an embryo where sperm RNA from trauma was injected, this offspring has symptoms of trauma, and also the further offspring. So, which is evidence that RNA, which is in sperm cells, they can carry messages of traumatic experiences and be responsible for the transmission across generations. Our research has been mostly conducted in mice, but we are, of course, very interested in translating it and having evidence, collecting evidence that some of the things we find in mice are also relevant for humans. So a couple of years ago, a former PhD student who then did a postdoc and now left the lab, and he's in Warsaw in Poland with his own group, Ali Javaid, who is from Lahore in Pakistan. He's a medical doctor trained in psychiatry, and he proposed to go to his hometown and establish a cohort, have contact with SOS Children's administration and establish a collaboration with the SOS Children's villages. And he established a cohort. He went there for 3 consecutive years for a couple of months. And he selected children based on the fact that they have lost their father and they were separated from their mother. So they were left in the orphanage by their mother because of the loss of the father and her inability to take care of the child, which is quite comparable to the paradigm in our mice. And Ali had these children. Now we have a cohort of about 100 children with control children who go to the same school but are with their family. They have not experienced any trauma. And he collected blood and saliva and had these children fill out questionnaires to ask them how they were feeling, whether they had fear, they had anxiety, and compile all this data. And it happens that all the children who are in this orphanage, they are either depressed or anxious or have a number of psychiatric, they are very fragile children. And we found by analyzing the blood and saliva in our lab in Zurich, exactly the same way as we did in mice, that the metabolic alterations we had observed in mice are also present in these children. What Ali did also was to collaborate with a diagnostic lab in Lahore to get sperm from men who were also traumatized. And we confirmed that some of our results in mice showing that RNA populations are altered in the sperm of traumatized males, that this is also the case in men who had been exposed to trauma before the age of 17. This type of work is quite difficult. It takes a lot of time, and collecting samples from humans with cohorts which are well defined and where we have all the information is huge amount of work. And it's not our specialty because we are experimentalists, but with this SOS Children Village study, we have started to confirm that our results in mice are relevant for humans. But we hope to be able to do more in the future. So what are the perspectives of this type of research for humans? We hope that it will help better understand how environmental factors can influence our cells and organisms. So to distinguish the innate features, phenotypes from acquired features, so those which are features that we inherit from our parents, from the features which we acquire throughout life depending on our life experiences. And by identifying epigenetic factors like RNA, which can be measured in blood, in saliva, in many different fluids very easily, that we could have use them as biological markers to be used for diagnostic or even one day for therapeutics. But if, let's say, we would be able to have a list of RNA, there are small RNA which can be measured very easily. They are called microRNAs. We have a list of microRNAs which we know are altered systematically by traumatic experiences in mouse blood. If we would have a very reliable list like this that we could be tested in humans, then it could eventually be used as a diagnostic to say, well, there may be signs of prior trauma in this person, which may make this person more susceptible to develop cardiovascular disease or psychiatric disorder, and be of use for medicine. For evolution, maybe we can discuss this question, but obviously, if now it modifies the genome, not by changing the code, but by changing the epigenome, this can have quite important consequences for evolution. I'll just finish with a piece of art by this artist, Sandrine Dognaud-Renaud, who is a French artist, and she watched a YouTube video of one of my conferences I gave many years ago in Paris, and she was very inspired. I think for her family story, she was very interested in the concept of transmission of life experiences or traumatic experiences across generations. And during the confinement, actually in 2020, she built this piece of art which reflects ancestors that she disposed in the form of a helix in this nice metallic frame. So the sculpture has a little story because it was exposed in different places, and last February it was for a couple of days in the Grand Palais in Paris. And in the field of epigenetic inheritance, there is one pioneer whose name is Michael Skinner, because Michael Skinner is the first person who showed that endocrine disruptors, pesticides like vinclozolin or methoxylor, who are used in agriculture, mostly on vineyards, that these compounds can have effects across multiple generations. And Michael Skinner is teaching in Paris every year, and he happened to be there in February, and we both were at the vernissage of this exhibition in the Grand Palais, so that was a nice moment. And I decided actually to buy this sculpture for the lab, and it's soon going to be installed in our institute. And for those of you who are interested, you can visit us to see this very beautiful piece of art. I thank you for your attention. I'm ready to answer any questions you may have.